In this video, we're going to talk about the basic prokaryotic structures. Now, prokaryotic cells, they are microorganisms, but they are single cellular organisms that can be found in two different domains, the archaea and the bacteria. We're not going to look too much at that archaea domain because they're not going to lead to human diseases. We are going to concentrate on the bacterial domain because those are going to have the prokaryotes you're used to seeing, the E. coli, the strips, the staph, etc. Now, as we go through the video, a couple of things to point out. Recognize that some of the features or structures are going to be found in all bacteria because they are cellular organisms. So it's going to have a cell membrane. They're going to have cytoplasms and ribosomes and even a cytoskeleton. But there's going to be a lot of those structures that are going to only be found in some bacteria, or depending on if they're even found, their look is going to vary depending on their type or the group that they belong. Those are going to be like their cell wall, those appendages of the flagella and the pili, and even some of their internal structures. So to begin this discussion, we're going to take all these structures and features and we're going to break them down into three categories: the appendages, the external structures, and the internal structures, and that'll help us with organizing and learning the parts. So let's begin with the appendages. There are two main appendages: the flagella, seen here as that long green tail-like structure, and then there are pili. Here pictured in this are the fimbriae pili, but there's also a pili known as the conjugation pili. Let's start with the flagella. Now, flagella, like in the eukaryotic cells, which you may be more familiar with, it is used for movement. But unlike the eukaryotic flagella, which is a back and forth wave-like motion, think about what happens with the sperm. In the bacterial cells, the flagellae turn in a circular motion. So they go clockwise or counterclockwise instead of that back and forth wave-like motion. Now, what allows for that is this structure of their flagella. Their flagella have embedded within their cell envelope, they have this thing known as a basal body, and it's consistent of a rod and different number pairs of rings, but then it is that shape there that allows it to rotate or twist within that cell, the cell envelope. You can see here the filament, which is the piece that you would see external to the cell. It's then attached to that basal body through a hook, and that whole structure rotates or turns. Now, if a bacteria has a flagella, because again, this is one that is found on some, but not all bacteria, the arrangement can vary. So here you see in letter A that we have one flagella coming off of one end or pole, and that is known as a monotrichous. In B, you see that there are flagella on both ends, and so we call that amphitrichous. In C, you have the flagella coming off of one end or one pole, as with A, but the difference between A and C is A is one flagella, and C, there are multi-flagella coming off of the end. And then in the case of D, we refer to that as peritrichous because those flagella cover the entire body of the bacteria. No matter what arrangement it has, flagella are still going to move in that circular motion, and they're still going to be used to propel the bacteria. Now, the other appendage that we discussed is the pili. The pili are not as long or as prominent as the flagella. They're going to be thin protein tubes that are attached to the outside of that bacteria. Now, not all bacteria, again, have pili. And when you do find them, it's most likely going to be found on a gram-negative bacteria. Pili are not used for motion. They are used for adhesion or to attach the cell. So in the case of the fimbriae are short little pili found across the entire cell. They are going to be used to help adhere a bacterium to surfaces and to other bacteria. Think of them, if you will, as like Velcro, okay? So just be cautious because again, that when it is a pili and bacteria cell, even if it's just on one end and looks like cilia, these are only going to be used for attachment. There's no motility about them. The conjugation pili, sometimes referred to as the sex pili, it allows for the two cells to adhere to one another, so it's got adhesion, and it's then going to allow the cells to pass a piece of DNA known as a plasmid from one cell to another. Conjugation pili we will discuss a little bit further when we get to the genetics. Now, those were our appendages. Let's talk a little bit about our external structures. So our external structures, we have two main structures. We have what's known as the glycocalyx, seen here in red. And see how it looks as a capsule? We're going to refer to those different structures or forms for the glycocalyx. This one here happens to be a capsule, so it's actually labeled as a capsule, but that is the glycocalyx. And then we have the cell envelope, which is going to have the outer membrane, the cell wall, and the cell plasma, okay, or cell membrane. The only one that will be found for sure in all cells is going to be that plasma or cell membrane. The cell wall and the outer membrane will be dependent on which type of cell it is. Let's talk a little bit about that glycocalyx then. So it is a glycocalyx because it has polysaccharides, which you may recall, that means it's sugar. Okay, so it's got a sugar coat, and this sugar coat is going to be found on the outside of the cell. It's made on the inside and then it's excreted and put to the outside. The glycocalyx will have one of two structures. As you saw in our picture, the red structure there, we call it a capsule because it is a thick, well-structured unit and it's going to be firmly attached to the cell. On the other hand, there can be a thin, loose layer that's really unorganized. It might look more like mucus, but it's actually not mucus, and it is called a slime layer. So the glycocalyx or sugar coat, two different looks to it, can be a capsule or a slime layer. Now, the cell envelope is the second of the external structures. The cell envelope can be made up of up to three pieces. It can have a cell wall, it can have an outer membrane, and all cells though are going to have that cell membrane or that plasma membrane. So let's talk a little bit about that cell wall component. Most cells have a cell wall. Not all, but most cells have the cell wall. And we can categorize our cells or group our cells or bacteria based on their type of cell wall. So we have three main categories. We have a gram-positive, a gram-negative, and an atypical type of cell wall. So if it is a gram-positive cell, it's going to have a thick cell wall composed of something known as a peptidoglycan. If it is a gram-negative, it's going to have a thin layer of that peptidoglycan, and it's going to have an outer membrane. So then what is this peptidoglycan? Peptidoglycan is going to be the main component of bacterial cell walls, okay? And what peptidoglycan is made up of is sugar and that are linked by protein bonds, okay? The two sugars are glycans, the N-acetylglucosamine, which we call NAG, and the N-acetylmuramic acid, which we refer to as NAM. Those sugars are linked to each other, and then we take those chains of sugars and we link them through peptide protein, if you will, cross links, and it cross links at the NAM sugars. Now there is a whole separate video talking about peptidoglycan and cell wall structures, so look for that as well. But here's just kind of an overview. The top is the gram-positive, and it has what we refer to as a thick cell wall. So you see here blue is that thick peptidoglycan sheet, okay? And again, it's going to be made from lots and lots of peptidoglycan sheets pulled together. It gives you a thick cell wall, which is, as you can see, to the outside of that cell membrane represented here in the gray. Now in gram-negative you have the cell membrane again in gray, and notice how the blue, the peptidoglycan, is very thin compared to up here in the gram-positive. But you notice it has an orange layer, that is the outer membrane, okay? And in this cell envelope structure, the gram-positive has two components. It has a cell membrane and it has a thick peptidoglycan cell wall. In the gram-negative, there's three components. It is composed of all three components: the cell membrane, there in gray, the cell wall in blue, and then an outer membrane in orange. Now, I said there's three types of cell walls, or three categories, if you will: gram-positive, gram-negative. The third category is the atypical cell wall, and those are going to be ones that are not made up of peptidoglycan or not possess a cell wall at all. So acid-fast bacteria, they have a cell wall, but their cell wall has a unique substance known as mycolic acid, which is a wax embedded within it, and so it gives it this thick waxy cell wall. So it's about the same thickness as gram positive, but it's got mycocalyc acids. And this is going to be the category for your mycobacteriums like tuberculosis. So the acid fast are often going to be pretty severe pathogenic bacteria. Then you've got the other group, the mycoplasmas, which do not possess a cell wall. And that is how they are atypical. So gram positive, gram negative, and atypical for your cell walls. Now, all cells possess the cell membrane. When you talk about a bacterial cell membrane, it is the basic phospholipid bilayer with the embedded proteins, that fluid mosaic model that you have learned before. Bacteria are going to have that same cell membrane structure. Now, where they're going to vary from the eukaryotic cell is in the function of that cell membrane. Now, just like in the eukaryotic cells, it is going to be a selective barrier, what comes in and goes out. But because those prokaryotic cells do not have organelles like eukaryotic cells, the cell membrane serves for all those functions that you may have remembered in your eukaryotic cells that the cell organelles would perform. So in the case of the prokaryotic cell, the cell membrane is going to help with ATP or energy production. It's going to help with protein synthesis, synthesis of cell wall components, DNA replication. It's going to have that flagella is going to be embedded in there. It's going to help the cell to know a little bit about its environment through the chemotaxis and sensing what nutrients or toxins might be present. So the cell membrane, even though it has a very similar look with that phospholipid bilayer as a eukaryotic cell, it's going to have a lot more functions. Now let's talk about the internal structures of a bacterial cell. So when we talk about internal structures, we're going to have a nucleoid, okay, which is going to be its DNA, if you will. We're going to have plasmids, which are also going to have some genetics, the ribosomes, the inclusions, and the endospores. Now, these are internal structures. Notice I did not call them organelles. They are not organelles because they're not bound by membranes. But we're going to look at the first three bullet here because those have genetic material associated with them. So bacterial chromosomes, and it's not like a eukaryotic cell where you have a nucleus and pairs of chromosomes, etc. In bacteria, I'm going to flip back here, it's one large circular chromosome that has 500,000 to 10 million nucleotides about it. It is going to be that double helix DNA structure, but it is going to be a circular chromosome. It is going to be twisted and compacted and put together in a very concise, neat little area that we refer to as a nucleoid. It is not a nucleus, it is a nucleoid. We will look more at the bacterial chromosomes when we get to the genetics. Now, all bacteria are going to have the chromosome. Some bacteria will have a plasmid, and a plasmid is a small, round, circular piece of DNA with only a few non-essential genes on it. But why the plasma is so important is because generally if you find a plasma in a bacteria, it's often going to be a pathogenic type of bacteria because usually those plasmids are going to have a virulence factor. For instance, it's going to have something that allows the bacteria to be resistant to an antibiotic, or it's going to allow a bacteria to produce a particular toxin. And so the plasmids are going to be important genetic features, but again, it's not found in all bacteria. The final genetic structure is the ribosome. So ribosomes are found, of course, in both eukaryotic and prokaryotic cells. They are going to be a site for protein synthesis. But the difference between the eukaryotic and prokaryotic cells is the way that they are put together and then their size, if you will. In the bacterial ribosomes, you're going to have two subunits, one which is 30S, one which is a 50S, and that gives an overall size of a ribosome of 70S. Okay, it's not an addition, even though you might want to do that, but 30 and 70 and it are the two subunit sizes, and it totals to give an entire ribosomal unit of 70S. Other internal structures, again, not organelles, but structures. Some bacteria are going to have a structure known as an endospore. An endospore is a form of survival. It is not found in all bacterial cells. It's generally found in the Bacillus and the Clostridium genus of bacteria. The endospore is going to be when they take their genetic material, wrap it in a very thick protein coat, okay, known as a spore coat, and then it allows that bacteria to go dormant when environmental conditions become very harsh and not suitable for the bacteria to live. Then when the environment becomes suitable again, viable if you will, then that endospore will turn into a viable bacteria, and then the bacteria will start to reproduce, etc. So endospore is for survival, not reproduction. And then bacteria will have inclusions. The inclusions are just going to be areas where the bacteria is going to contain various nutrients or substances needed to help with day-to-day life or survival. And again, what the inclusion body's function is going to be based on what that nutrient is found within it. Okay, so not all bacteria have endospores, or inclusions are going to vary by the bacteria. But that is the basic structure of the prokaryotic cells. Again, look for the video on the cell wall for more details on all of how that is structured and how it functions.